Okay, Chapter 22, Infectious Diseases of the Gastrointestinal System. The gastrointestinal tract, or GI tract, is essentially one long tube which starts at the mouth and ends at the anus. We ingest foods and liquids through this system. They travel throughout and then we release waste at the end. So it is open in two places to the outside. It has constant input of external microbes. So for this reason, it's very heavily protected and monitored to prevent infection. The oral cavity, or the upper part of the gastrointestinal tract, is protected by saliva and lysozymes that are antimicrobial. We also have secretory immunoglobulin A, which is the antibody secreted by all the mucous membranes in the host, the human host. And then there's presence of several groups of normal microbiota, which protect the oral cavity by microbial antagonism. The rest of the gastrointestinal tract is closely associated with lymphoid tissues, also called GALT. And these lymphoid tissues are responsible for monitoring the presence of pathogens in the gastrointestinal tract. If pathogens are detected in the food or drink we ingest, then the immune system can be alerted by these lymphoid tissues. We have secretory immunoglobulin A released in the lower parts of the gastrointestinal tract, and then the presence of a rich group of normal microbiota, which is variable from person to person and has effects on everything from overall health of the person to their body mass index to their mental health and predisposure to diseases like depression. The first of the gastrointestinal infectious diseases we'll discuss is gastritis or gastric ulcers. So although these can be caused by a number of different factors, we're going to focus on those caused by infection with Helicobacter pylori. And gastritis or gastric ulcers are inflammation of the gastrointestinal tract and sores on the stomach or upper portions of the intestine, most of the small intestine. So Helicobacter pylori is a gram-negative rod. It colonizes the stomach of an estimated half of the world's population and has evolved over 100,000 years to coexist with humans. Its mode of transmission is probably by fecal-oral or oral-oral route, where it's often seen that there will be familial infection if one member of a family has Helicobacter pylori in their gastrointestinal tract. It's likely that other family members will as well. The main virulence factors of H. pylori are its ability to adhere very strongly to the epithelial cells that line the stomach, and production of urease, which is essential for converting urea to bases that can neutralize the acidic pH of the stomach. A large portion of the damage caused by Helicobacter pylori infection and resulting gastric ulcers is due to the inflammatory response by the host's immune system. The main tissues infected are those in the stomach, and there's a number of signs and symptoms associated with gastric ulcers, including stomach cancer, which can result from long-term infection that goes untreated. Diagnosis is done by inhalation of radioactive urea, which in the presence of Helicobacter pylori and urease produced by the bacteria will be converted into radioactive carbon that will then be exhaled by the person. So when the inhaled urea is then resulting in an exhaled radioactive carbon, the person is positive for Helicobacter pylori infection. There are also ways to test for Helicobacter pylori antigen in stool samples. Currently, there is no vaccine. The way to treat Helicobacter pylori infection is over-the-counter medicine for symptom relief and antibiotics to clear the bacteria. So the tissues infected by this bacteria are largely the stomach. And the main difficulty of infecting the stomach is that it has a very low pH with a lot of acidic molecules to help in digestion of food. So illustrated here are the 4 steps that Helicobacter pylori goes through in order to become established and infect the stomach lining. So in the first step, Helicobacter pylori, which is got flagella, which allow it to be motile, will attach to epithelial cells that line the stomach layer. To do that, it has to bore through a protective layer of mucus that is in between the stomach lining epithelial cells and the gastric fluid, which is really acidic in pH and is important for breaking down food. So it bores through the mucus layer, then Helicobacter pylori attaches to the epithelial cells. Helicobacter pylori often produces urease, which is an enzyme that can take urea present in the stomach environment and turn it into a base, which will then neutralize a small area of stomach acid so that the H. pylori can exist in this neutral pH. When it establishes this area of neutral pH, other Helicobacter pylori can come to the same spot. And the inflammation that results from the presence of Helicobacter pylori in these tissues will start to degrade the mucus, and the epithelial cells are then in direct contact with the gastric fluid that has a severe pH. Not severe pH, acidic pH. So as a result of this contact between gastric fluid, epithelial cells, and the immune response, we get extensive damage, death of these epithelial cells, and you get sores that can then start bleeding and cause a lot of pain. Okay, the next of the gastrointestinal tract infections is Salmonella enterica, which is a gram-negative enteric bacteria. Enteric means that it targets the gastrointestinal tract specifically. There are a number of different types of Salmonella called serotypes, and these are based on differences in their cell surface antigens, which have slight variation between different serotypes. Salmonella enterica contaminates estimated 10% of chickens destined for human consumption because it is a part of the normal biota of chickens and cows and some reptiles. Salmonella enterica can also be present in or on eggs, and it's estimated there are 1.2 million infections reported each year and approximately 500 deaths. Depending on the serotype, Salmonella can cause typhoid fever, which is a very severe disease, or it can cause the more milder salmonellosis, which we would probably just call food poisoning if we contracted it. The modes of transmission are direct contact via kissing, vehicle via food, and mechanical vector via reptiles, rodents, poultry, or cattle, which may have the bacteria on the outside of their bodies. So if you handle these, you then could have the microbe on your hands and ingest it yourself. Virulence factors, it has a great ability to adhere to host tissues. It produces proteins that help evade immune defenses, and it has the LPS endotoxin, which causes a severe immune response. All of the gastrointestinal tissues can be infected by Salmonella enterica, and there's a list of signs and symptoms. Diagnosis is only done in very severe cases and can be done from a stool sample. So the ways to prevent typhoid fever or salmonellosis are to avoid contact. There is a vaccine for poultry or for travelers to areas where typhoid fever is endemic. Treatment will be fluid and electrolyte replacement, and for very severe cases, antibiotics. So with Salmonella enterica, if it is of serotype that causes salmonellosis, that will be Salmonella enterica serotype enteritis, which is found in mice, or Salmonella enterica of the serotype typhimurium, which is found in chickens. So this disease caused by these two serotypes is self-limiting, and after a few days, the person will feel better. There is a serotype called typhi, which is present in humans only, and this can cause the much more severe systemic disease called typhoid fever. Some people are carriers of Salmonella enterica serotype typhi, and the most famous carrier of this bacteria was Typhoid Mary, who was a cook in New York in the early 1900s and is thought to have infected dozens of people because she carried Salmonella enterica serotype typhi without any symptoms. Prevention of typhoid fever is mostly to avoid eating the outside of food, so this could be the eggshell or the peel of a banana, to cook foods to the recommended temperature, or to boil things in order to kill the bacteria that might be present. And then there is a vaccine available for people traveling to areas where typhoid fever is endemic. The last of the gastrointestinal diseases is cholera, which is caused by the gram-negative bacteria Vibrio cholerae. It is a common-shaped gram-negative rod. It has flagella, and there have been several outbreaks of cholera after natural disasters. And this microbe has shaped human history because at points in time, half or a significant part of the world's population has been wiped out by cholera epidemics or pandemics. The mode of transmission is vehicle, so you get cholera from food or water that contains the bacteria. It spreads most easily in warm, saline conditions, and it often occurs after natural disasters where sewage will mix with drinking water. So if you have cholera present in the sewage, it mixes with drinking water and then is ingested by everyone that drinks the water, and then you have a cholera outbreak. The flagella of cholera are important for motility and also to penetrate the mucus barrier of the small intestine. The bacteria can also adhere to intestinal epithelial cells and multiply, but it never enters the host epithelial cells or deeper tissues. It stays on the surface of the gastrointestinal tract. And then the bacteria produces a cholera enterotoxin, which is a toxin that targets the gastrointestinal tract. This toxin disrupts the normal physiology of the intestines, so large amounts of electrolytes and water are shed by the host. So there's massive diarrhea occurring when a person contracts cholera. In addition to vomiting and massive diarrhea, there's a characteristic appearance of the diarrhea, which is called rice water stools. And this is because the volume of fluid voided by the host is massive. There's also flecks of mucus in it, which give it the appearance of rice water. Because of the severe dehydration as a result of cholera, there are associated muscle cramps, severe thirst, flaccid skin, and sunken eyes, and if untreated, will kill the host within 48 hours. Diagnosis is done in cases where it's essential to identify the source of the cholera outbreak in order to contain it. So from stool samples, the bacteria can be identified by microscopy, and you can detect antitoxin, which is a response of the host's immune system to the cholera toxin being produced in the blood serum. And then to determine and track the source of infection, we can do PCR to find Vibrio cholera DNA and sequence it so we can identify where it's coming from. Prevention is by proper sewage treatment and water purification, so ensuring that drinking water is safe. And detection and treatment of asymptomatic carriers to ensure that they aren't infecting others unknowingly. There is a vaccine available for those in endemic regions or for people traveling. And treatment is oral rehydration therapy, which is just replacing the large amount of fluid loss, and oral antibiotics. This is what Vibrio cholera looks like. So it's described as being comma-shaped because it's partially curved, and it's going to infect the small intestine. And the current 4 years old countries reporting cholera outbreaks are shown here. There are cots designed for cholera treatment with an area where the diarrhea can exit the person, so you have a literal hole in the cot. And then on the right is Ringer's lactate, which is a solution to replace electrolytes and fluid for the fluid loss as a result of infection. That concludes chapter 22.